Master in Anatomy and Physiology with Contd.
Guys, this is my next lecture. It's also about cell membrane. In last lecture, we briefly talked about the structure of the cell membrane or phospholipid bilayer, and we talked about different proteins that are embedded with the membrane, and they are like helping to transport different types of substances and molecules. So, but in today's lecture, what we're gonna mainly focus is the types of transport we're gonna see, which may be active transport or which may be passive transport. I'm going to focus on those things in depth. Okay, so that's the main goal of the today's lecture. So let me start with that. So I have some note in back of the whiteboard you can see here, and it's going to be very helpful to understand those things. Let me show you these things. In cell membrane, you basically have usually you have passive transport and the active transport. The passive transport, you don't need any energy. So I crossed out the E, which is energy. You don't need that. On the active transport, you need energy. You have to utilize some ATP to perform work, okay, to push molecules against their will. We're trying to push them, okay, against the concentration gradient. That's why active transport needs energy. Remember that passive transport works on the concentration gradient. From the high concentration to the low concentration, the concentration of solute. Okay, I'm not talking about water, I'm talking about solute. The solution may be hypertonic, hypotonic, or isotonic. As I mentioned somewhere here, hypertonic, isotonic, and the hypotonic. What is hypertonic means? It means the solute concentration is more. Okay, isotonic means it is in between. It is not hypo, it is not hyper. And the hypotonic means the solute concentration is less, but you have a water concentration or solvent concentration is more. So that we call a hypotonic solution. So what happened on the passive transport, it goes from, okay, it goes the molecules like where the hypertonic solution is, what will happen is from the hypotonic, where the water concentration is more, the water gonna move toward the hypertonic solution because there are more what? Water. It trying to get in. Let's water come inside. Okay, hypertonic solution trying to always let water to come inside because it's hypertonic. Okay, that's why it is very important to understand. And the diffusion and osmosis usually, okay, these are the same idea but like a little bit difference on those. And the osmosis is a type of a diffusion basically. In the case of diffusion, what will happen is moment of what? Any chemical mixture, any chemical molecule, okay, they're gonna move. But in the case of osmosis, the water will move. Diffusion can take place in any direction. For example, you spray a perfume in a room. It's going to diffuse through the air and it can travel to the different directions. But the osmosis, you need the membrane, the semi-permeable membrane, so the water can pass through it, right? So you need the membrane, so osmosis requires membrane. That's a very important thing to remember there. So that's the basic difference, okay? So next one is osmotic pressure. During the travel of the molecule through a membrane, they give a pressure to the membrane. That pressure is called osmotic pressure. And the membrane may be artificial or which may be a natural or cellular membrane, right? So that's the important thing. And as I told you, the concentration gradient of sugar solution, let's say, if we have a container here and you have a like membrane right here, right? And what, let's say, you have some sugar solution here, where you have a concentration of sugar is more, and here the concentration of sugar is less, but the water concentration is more here, right? So this is a hypotonic, this is a hypertonic. What will happen is the water gonna move this side. okay, water gonna move toward the hypertonic solution side. So this is a very good example of the osmosis, okay, through a semi-permeable membrane. And the osmoregulation is another term you need to understand right here. Osmoregulation is a control of solid concentration and the water balance. For example, in the case of a protist or paramecium caudatum, okay, what it happens is the it usually loves pond water, right? It usually in the pond water, the outside environment, the solution pond water is hypotonic usually. The inside of the paramecium is hypertonic. So what will happen is water gonna go inside the paramecium so fast, the rate is really fast. But paramecium has a natural control on that because it has a contractile vacuole and it just like push the water out of the cell also. So that's how the paramecium performs osmoregulation. Sometimes the diffusion may be facilitated diffusion in which you need some type of help. You're going to get some type of help from channels, but you're not still using energy. You're not using energy, but you're just getting the help of the channels, which may be the proton, which may be the sodium potassium pump, or which may be a proton pump. Okay, so these are the channels you may use. You need help of the transport proteins to transport the things, but as I told you, you don't need any energy. There is no expend of energy, so that's why it is called a facilitated diffusion. Okay, so no energy, but you just need the help. Okay, for example, you have some water channels in many cells of the kidney cells and the RBCs. So what will happen is the water channels will allow to pass, okay, different ions also, or different types of solids, right? So it will allow to pass different types of substances inside. So the channels that allow water to pass are called aquaporins. We need to understand that. Sometimes you may have ion channels also. These ion channels works as a gated channels. They turn on and turn off. Okay, they allow to pass the ions inside. For example, in the animal cell, what will happen is there is a sodium potassium type of channel. Okay, like pump in human body, right? What will happen basically is the pumping of sodium and the potassium across a membrane through against a concentration gradient is usually performed by the sodium potassium pump. But in the case of fungi, plants, and bacteria, they may have proton type of channel where they pump the H plus ions across a membrane, right? And another thing is like you may have carrier proteins sometimes. The carrier proteins, for example, glucose transporter is a type of carrier proteins. What it does is it carries proteins, okay, and what it does is undergo like change or transformation, and it will change the inner side, whatever inside the inside compartment. What it does is uh, it changes the environment of the inside part. So that's the carrier proteins does. So that's carrier proteins very important also, right? And another thing I want to mention is sometimes you need to transport like bulk of the materials, like huge molecules, right? So what happen? You may have different types of mechanism to do that. You may have exocytosis, you may have endocytosis. Exocytosis is a fusion of vesicles with a plasma membrane. Okay, it is a fusion of vesicles to a plasma membrane and transporting. Okay, transporting that whatever substance on that to the outside. Okay, you just like throwing out, like exocytosis. Okay, you form a vesicle and you're throwing out. In the case of endocytosis, you forming a vesicle and you're draining inside. That is the endocytosis. And the endocytosis goes through like different ways. You may have phagocytosis, you may have pinocytosis, or you may have receptor mediated endocytosis. In a phagocytosis, it's like engulfing all of it. Okay, it is eating. Okay, pinocytosis means drinking cells like that. Okay, and the receptor mediated means like you have a certain type of receptors. They will allow to transport like huge like macromolecules or those like in a huge amount. Okay, so these are the ways how different molecules enter and exit from the cell membrane. And remember that in the case of cell membrane, there is a sometimes there is a voltage difference on the in the case of the membrane. What I mean by that is like voltage difference is means like there is a electrical charges across the membrane. Sometimes what will happen is in the case of membrane, most of the membrane, the inside part is negative and more negative than outside is positive type of thing. And what will happen is the ions which are positive, they rush inside, and some of them which are negative, which run outside of the membrane. So there is a voltage, there is a ranges in the membrane potential, right? So that is a voltage across a membrane, which is usually called a membrane potential. You may see that, which may range from negative 50 to 200 millivolt. Okay, so this is why it is very important. So diffusion is usually happens through two mechanisms, which may be due to chemical force or which may be electrical force. That's why the diffusion totally we consider as a electrochemical gradient type of moment of diffusion across a membrane. So this is what we cover today. In next lecture, what you're going to cover is you're going to cover the different components of the cytoplasm or different organelles which are lying, which are just like hanging in the cytoplasmic area or cytoplasmic compartment. Thank you for watching. See you. Bye-bye.
Please subscribe to see more videos regarding anatomy and physiology.